Well, good morning, everyone. My name is Kendra Phelps, and I'm a senior scientist at EcoHealth Alliance. And today I'm going to be talking about the what, why, and hows of collaborative research networks, and then share with you some of my experiences and lessons learned while coordinating field and lab activities for the Western Asia Bat Research Network, which is a regional surveillance network focused on bat-associated coronaviruses. So there's no one succinct definition of a collaborative network, but some key points are expressed in these three definitions that I'm going to show you. The first being that researchers who are committed to a common purpose, approach, and goal, and hold themselves mutually accountable. So in other words, every member of the network is equally committed to the outcomes of the network. Collaborative networks arise from needs, interests, and questions of researchers who are most invested in the findings or outcomes of the network. My favorite part of this definition is research is conducted with rather than on. So this really highlights the collaborative in collaborative research networks. And lastly, collaborative networks lead to research outcomes that far exceed that could be achieved by individual researchers. And collaborative networks are strongest when they're engaged with experts in diverse disciplinary fields, which I think we all know this and we've heard it multiple times. And what's relevant to this workshop is when we're incorporating One Health research approaches, collaborative networks require knowledge exchange among experts in animal and human health, wildlife ecology, virology, sociology, environmental scientists. I could go on and on and on, but as you can see, it's quite a long list. But the key point is they should be collective and collaborative networks should be multidisciplinary. And collaborative networks often have their beginnings at the national level, but several advantages to coordinating at the regional level include the ability to take effective action on things such as shared viral threats that span geological or geographical and temporal scales. So, for example, viruses with mobile hosts, let's say bat species, can be easily spread across borders. And this requires regional collaborations to ensure that there's reduced risk of spillover and spread. So without regional collaborations, surveillance efforts in one country or in one network node may be insufficient to detect viral threats from a species elsewhere in its range in another node in their collaborative network. In general, collaborative networks have three main objectives. The first being to build social capital. So this can range from shared resources such as reagents and lab equipment to development of field and lab protocols. There's skills transfer, so this is hands-on training in those field and lab protocols. Or even due to the pandemic, we've gotten very creative with virtual training workshops. Many of you have probably participated in some of those. And then there's also networking to build connections among the network members. And that's what we're here for today. Collaborative networks strengthen the coordination of shared activities, such as field and lab activities of a bat corona surveillance program, so what I'll be talking about. So these shared activities are done in such a way that common effectiveness is enhanced. Well, that's to say that activities across a network are standardized so that they are comparable. And lastly, by combining the network's collective expertise and resources, so that social capital, with comparative data collected using standardized methods, so coordination, you can answer bigger and more complex questions and expand the breadth of your own research. So you're maximizing the impact of your research. One example would be early detection of a zoonotic pathogen by one member of the network could preempt mitigation efforts that limit the spread from a small outbreak to a larger epidemic that is affecting other members within your network. So now that I've covered the what, who's, and how's of collaborative networks, I want to use the Western Asia Bat Research Network, or WABINet for short, as an example. And WABINet is the first collaborative research network in Western Asia to take a multidisciplinary approach by integrating research from bat biologists, taxonomists, conservationists, with virologists, veterinarians, and public health experts to protect both bats and public health at the regional level. So it's multidisciplinary and at a regional level. So first, let me give you a little bit of background, because many of you are probably wondering, where's Western Asia? It's actually 20 countries in basically the Middle East, so it extends from Turkey in the north down through the Arabian Peninsula and over east through Afghanistan and Pakistan. And it represents really a unique regional mixing pot, if you will, of bat species from different biogeographical regions. So it brings species together from Africa, Asia, and Europe. And there are approximately 100 to 110 bat species distributed across the region. Yet, despite this diversity in bat species, Western Asia, along with much of Africa, has limited published research on bats, period. Anything about bats. In fact, half of all Western Asia countries have 10 or fewer publications on bat species. And similar to that lack of effort on bats, or lack of research on bats, there's also a huge gap in research effort on bat-associated viruses, and it's even more limited. In fact, a quarter of all the countries in Western Asia have absolutely no publications on bat-associated viruses. So to fill this gap in research on bats and their associated viruses, the WABINet was formed by EcoHealth Alliance in collaboration with multinational and multidisciplinary stakeholders from the region in 2018. And it has three main objectives. The first being foster regional collaborations and information sharing through annual workshops that are held in each of the partner countries it's rotated through each year. And it's difficult to build a collaborative network without face-to-face opportunities to connect with other regional experts. So we host annual workshops to discuss ongoing research projects and identify critical research gaps. The first workshop was held in 2018 in Georgia, and the following year it was held in Georgia. And each of these workshops were attended by about 40 participants from 10 to 15 Western Asia countries. But in the last 2 years, we've had a really large increase in the number of participants because they've had to hold the workshops virtually. So the plus side is, with virtual workshops, you can have a greater number of attendees from a larger number of countries. The downside is you lose that one-to-one in-person connection. And during the workshops, there are discussions to identify research questions and hypotheses that are relevant to the region. And then we develop standardized protocols so that we can test these hypotheses and get answers to the research questions. And then we also try to weave in some fun activities like PPE races, just to kind of instill the importance of biosafety, but doing in a fun manner. And then during one particular workshop, we had an open discussion amongst participants about the links between bat ecology and viral transmission. And this is, Professor Tigga Kingston, who serves on the steering committee for WABINet, and she's standing here with the bat. And this is the before picture. So after some intense discussion, with diverse inputs from participants with very diverse backgrounds. So think about the viewpoints of a virologist and how they may differ from a bat ecologist. This was the final output. And you can barely even make out the bat in the center of the picture anymore. So needless to say, this was a really fun exercise in knowledge exchange. The second primary aim of WABINet is to leverage this existing expertise in the area to improve regional capacity to study bats and their associated viruses. And we're focused right now just on coronaviruses, which for us was kind of lucky since we started in 2018, not knowing what was down the road. But we also wanted to identify potential bat-human interfaces where this may facilitate viral spillover events from bats to humans. So working in coordination with network partners, we developed standardized protocols to safely capture bats, collect non-lethal diagnostic samples, and also to characterize the sampled sites to gauge both bat-human interaction potential and levels of human disturbance. And then we conducted extensive hands-on training on these protocols. And so far, I estimate we've trained at least 75 individuals from 7 countries in things such as using mist nets and harp traps to capture bats, how to collect basic demographic information, which is typical of any ecological study. And opportunistically, we're also taking acoustic recordings, and this is with the goal of building the first regional call or acoustic reference library for Western Asia. And then also how to collect non-lethal diagnostic samples, which we need to screen for viruses. Or in the case of the wing biopsy punches, we use to confirm bat species identifications. And as I mentioned earlier, we developed standardized methods to characterize the sampled sites because there are vast differences in site conditions across such a large geographic region. So specifically at each of these sites, we're collecting data on environmental and human disturbance factors that may increase contact between bats and humans. So these sites range from a beautiful coastal cave on the edge of the Black Sea in Turkey. This cave receives little to no visits from humans, to a very active tourist cave in the canyons of Armenia. This is also a USAID-funded archaeological site. And also we documented at least 10 bat species also share this cave. And something very unique to this site that I've never seen before is it also, I keep forgetting to flip through here, has its own Wi-Fi signal. Something I've never seen in 20 years of studying bats. So let me know if you've ever encountered that before, but. And then we moved to the less glorious fish reproduction plant in Azerbaijan, where bats are roosting right in the roof that's overhanging the holding tanks that contain the fish as they grow and develop. As you can see from the 3 different sites I just showed you, in 3 different countries across the region, each has been exposed to a diverse gradient of interactions between bats and humans. So Tourist Cave in Armenia, where thousands of visitors are coming annually, or interactions between bats and, I guess you would say, livestock. In the case of fish at the reproduction plants here in Azerbaijan, to the pristine site in Turkey, where people are rarely visiting the site. We also wanted to strive for consistency in the collection and management of the field data across the regions. So we developed standardized data sheets. Doing so allowed for field data that was collected in a manner that was comparable across all of the sampling sites in the region. And then it was entered into EpiCollect by the network partners. And I'll come back to the importance of EpiCollect. So, and lastly, WABINET standardized field protocols ensured that all network members were aware of and complied with our animal ethics guidelines. So as John mentioned, IACUCs, and also biosafety regulations, which is the personal protective equipment, PPE, that he described as well. This is meant to protect not only the health of the bats, but also of the researchers. And to date, WABINET partners have sampled at 50 sites in 7 countries, and we really hope to expand our sampling efforts in the past few years. But unfortunately, we work in an area that is politically unstable. So countries like Syria, Yemen, Iran, Afghanistan have just not been open for sampling. Plus the pandemic really kind of put the stop on a lot of sampling efforts. And from these 50 sites, we've sampled nearly 4,300 bats of 37 species. So this is over a third of all of the bat species that are found in the entire 20-country region. So really good diversity of species that are being sampled. And from these bats, we've collected nearly 20,000 diagnostic samples, which are then sent to one of our 2 regional laboratory hubs, either in Georgia or Jordan. And transferring samples between using cold chain between network partners is no simple task, especially if you're working with World Courier. But I'll come back to that when I'm talking about my lessons learned. So working in coordination with the 2 regional laboratory partners, we developed standardized laboratory protocols to detect coronaviruses in fecal samples using standard consensus PCR and Sanger sequencing, as well as confirming bat species identification through barcoding. As I mentioned earlier, all of our laboratory activities are being conducted at 2 regional laboratories. So either the Richard Lugar Center in Georgia or the Royal Scientific Society in Jordan. And we did this because these 2 laboratories meet our biosafety and biosecurity regulations. We need at least a BSL-2 level lab to do our work, and they were able to comply with that. So this initial 2 laboratory system has really allowed for assurance of consistency across the molecular methods that we're employing, as well as compliance, again, with the biosafety and biosecurity regulations I mentioned. And lab training has taken 2 forms. One I like to call cross-pollination between the 2 regional laboratories. So this may be in the form of a one-day meeting that we hold a day prior to the annual network meetings when we were still having those in person. Now we've moved more to virtual training sessions, which are focused usually on a specific topic. Sometimes it's troubleshooting an issue, Other times it's focused on analysis of sequence data, such as bioinformatics. And in a few weeks, we'll be actually all meeting in Georgia. So people from the Jordan lab, as well as people from EcoHealth Alliance, will be meeting in Georgia to revamp and just kind of review our laboratory protocols to make sure that we're still being consistent and that we're using the best methods possible. Originally, this was planned for March 2020, but we all know what happened at that exact time, and so it's kind of been pushed off a little bit later than we like, and we've already processed about 60% of our samples, but it never hurts to reevaluate what you're doing and double check that you're being consistent in everything. And second, we aim to build the capacity of our network partners to conduct laboratory work through field to lab research exchanges. So network partners from one country may be collecting diagnostic samples and then typically sending them to one of the regional labs. But in this case, they're going with these samples and receiving 3 weeks of training on how to process their own samples at one of the regional laboratory hubs. So here we have 2 Omani researchers who've traveled to Jordan with their Oman collected samples to learn how to use the WabiNet standardized lab protocols to screen the samples for coronaviruses. And similar to the field collected data, we also strive for consistency in how we collected and managed the data across the 2 regional labs. So we developed a standardized data platform using what's called Airtable. And this is to ensure that the relevant data fields were captured and outputs from the labs were comparable between the labs. And both of the databases I mentioned previously, so EpiCollect for field collected data and Airtable for lab collected data, are merged together into a WabiNet specific relational database. And this is done via a unique animal ID that is assigned to every piece of data. So it's associated with an animal, and that allows it to be collated, and then it can be shared. This country-specific data can be shared across the network with all partners. So taken together, so the host, ecology, site conditions related to bat-human interfaces, coupled with screening diagnostic samples in the lab, will ultimately allow us to achieve our 3rd primary aim of WabiNet, which is mitigating or ideally preventing any zoonotic disease emergence through risk assessment modeling, while concurrently promoting bat conservation by identifying primary threats to bat populations across the region. So there's the agency part of the work. So I hope I've demonstrated how WabiNet is a prime example of a collaborative network that is regional and multidisciplinary in scope and strives to meet those 3 objectives of a collaborative network: to build social capital, strengthen coordination, and use social capital and coordination of the network for agency. So we're to have a greater impact as a network versus as an individual working alone in one country. So now I want to dive into some of the lessons I've learned in the past 5 years as a coordinator for WabiNet. Oops. So this lesson is pretty straightforward. Be a good collaborator. I mean, would you want to work with you is really the question. But in all seriousness, this boils down to involving all network partners from beginning to the end of the project. So from developing research questions that are relevant to your region, to implementing the project across the region, in a coordinated fashion to publishing the results together. But if you think back to one of the definitions given at the beginning of the presentation, researchers within a collaborative network hold themselves mutually accountable. So if a network partner is not fully engaged, this should be addressed early on, as it can have compounding or create issues down the line, such as future conflict or even resentment amongst network partners. So I've heard. Luckily, we haven't had this issue with WabiNet. Everyone's fully divested, so be a good collaborator. That's the foundation of having a great collaborative network. Data sharing. This is obviously critical for a collaborative network to function properly and also to achieve the ultimate goal of agency. But it can be difficult to balance the need for transparency and data sharing between partners with the importance of data privacy. So for example, individual partner countries may not want to disclose sensitive information about viruses that are found in their country. So I recommend developing data sharing agreements early on and establish what you're comfortable with sharing between partners within the network. And that's particularly important when it comes to publishing your findings. Another lesson learned is broad engagement. So 2 prominent ways of engagement that enhance network sustainability. The first being is engaging with regional policymakers to effectively communicate what is the relevance of the network's research and encourage, hopefully, sustainable streams of funding, ideally from different government entities. So for example, from the Department of Health and the Department of Agriculture and the Department of Environment or Wildlife Conservation. And another form of engagement is training the next generation. So this may be involving students or postdocs in the project. So examples for WabiNet include in-field trainings. So we involve students, a lot of university students are trained in the field by our regional experts on the standardized field protocols. And the second is what I was mentioning earlier, the field to lab research exchanges. These are designed to expose the network partners to every aspect of the multidisciplinary research project. So this from field data collection to lab methods, statistical and phylogenetic analyses. So if you put these together, these trainings will promote collaboration and creativity and provide participants with skills and perspectives needed to better approach a multidisciplinary One Health research. And honestly, training the next generation is critical. I love to do field work, but I've noticed as I'm getting older, it's much harder on me. So passing that on to the next generation keeps that knowledge exchange going and keeps these types of projects happening and building, and they bring new insight, honestly, to the projects. I think it goes without saying in results communication, it's important to not overplay or misrepresent your bat virus findings, especially given the potential consequences or threats that this may have to bat populations, as well as the sensitivity of the government or the public to such findings. Furthermore, publishing your findings in scientific journals only reaches a very specific subset of the scientific community, while community outreach, such as educational, which maybe Wanda, you mentioned this booklet as well. And I meant to share a link. If you just look up Living Safely with Bats, this is a great educational booklet created by the Predict Project. It's been translated in 12 languages already, and we hope to use it for the WabiNet project, but it details not only the ecological importance of bats and why we need to protect them, but also biosafety measures that the public can take to mitigate any kind of type of risk so that they can live safely with bats. So it's a wonderful example of how to translate your findings to those that are at actually the greatest risk of viral spillover, but they're just not aware of the risks or the behaviors and how they can change those behaviors in order to live safely with bats. And then lastly, I'm going to end with my favorite lesson that I've learned is just to have fun. This doesn't always have to be mundane work, if you will. So fun can take many different forms, and we'll so one example is when you're fully donned in PPE, get up and dance and shake off that nighttime fatigue. That's one of my favorite videos. Or jet lag, that's my permanent state of being, usually. So I use that energy, usually which kicks in late at night, like last night, so I have very little sleep. I was ready to go out and catch bats last night, so if anyone was ready, I was jazzed and ready to go. So I try to use that energy to pump up my collaborators when we're in the field. That or I terrify them. I can't figure out which one. Sometimes I work in not so secure places, so I have security guards that flank me everywhere I go, literally everywhere I go. So I have fun sometimes, and I make them take stupid, silly pictures with me, but that's my way of having fun. They might not think it's fun, but I have fun. Even during the pandemic, we found ways to have fun. We turned self-testing into a team-building exercise. So every 2 days in the field, we do self-testing, and I found that the teams are actually reminding me when it's self-testing day. So don't forget, we have to self-test today. So it's actually turned into a fun activity, if you can think of swabbing yourself as a fun activity. But for me, these little guys are what makes the work the funnest and puts a smile on my face, which I feel like it also puts a smile on their face too.